Good morning children, I am Isis. Now I am going to teach you translation or biosynthesis of protein. Translation is the process of synthesis of proteins from mRNA. Actually, the genetic informations are transmitting from DNA to mRNA, or we can say through mRNA it gets translated for the synthesis of protein. Okay, so the DNA transmit genetic informations to form mRNA. That process is called a transcription, and the process of synthesis of proteins from mRNA is called translation. So transcription we already learned. Now we can see translation in detail. And there are some factors which involve in the translation process. Which are the factors which involve in the translation process? We can see first. First one, an mRNA strand which is transcribed from the DNA. And this acts as a template. So mRNA strand which acts as a template, and in mRNA the translation starts from 5' end. Okay, 5' to 3' end. On mRNA the translation process gets started. And the second factor is the ribosome complex. Ribosome complex, which consists of a smaller subunits of ribosome and a larger subunits of ribosome. And the third one, tRNA, a charged tRNA with a particular amino acid. And the fourth one, a releasing factor or a release factor. So these are the major factors which involve in the translation process. Next we can see what are the steps involved in the biosynthesis of protein or translation. So the steps are, number one, initiation. So the first step is initiation. And in initiation there are some processes. The first process in initiation is the activation of amino acid. Activation of amino acid. And the second step is charging of tRNA. Charging of tRNA. Transfer RNA. And the third step is the formation of mRNA ribosome complex. mRNA ribosome complex are formed here. And the second step in the translation process is, as usual, elongation, extension of the peptide chain, polypeptide chain. And the third one is the termination process, ending of translation. So these are the major steps involved in the translation process. They are initiation, elongation and termination. In initiation we have to see the three steps: activation of amino acid, charging of tRNA and formation of mRNA ribosomal complex. Now we can see the initiation process in detail. So in initiation the first step is the activation of amino acid. Activation of amino acid. So the amino acids present in the cytoplasm get activated with the help of ATP and an enzyme. So the amino acid, with the help of or in the presence of ATP, an enzyme known as aminoacyl synthetase. Aminoacyl synthetase, it is an enzyme. So in the presence of ATP, the amino acids can activate with the help of an enzyme aminoacyl synthetase. Then it forms aminoacyl. Aminoacyl. So the activated amino acid is called aminoacyl. Activated amino acid is also known as aminoacyl. So the amino acids get activated with the help of the enzyme aminoacyl synthetase in the presence of ATP. Okay. And the second point is charging of tRNA. Charging of tRNA. So here in the charging of tRNA, this activated amino acid react only with the tRNA which is specified for a particular amino acid, and then it gets charged. So the activated amino acid get bind with the particular tRNA molecule which has a particular anticodon. For example, the tRNA with the UAC anticodon that bind with the methionine. So the methionine amino acid get bind with the tRNA which is having UAC as the anticodon. So we can say the tRNA with anticodon UAC bind with the amino acid methionine. So methionine when get activated with the help of an enzyme, it get bind with a particular tRNA which is having the specified or specific anticodon. Is it clear? So that is called a charging of tRNA. So we can write over here the activated amino acid bind with or react with the tRNA molecule specified for particular amino acid to form charged tRNA. Then it get charged. So this is the second step under initiation process, charging of tRNA. Next, third step under initiation process is formation of mRNA ribosome complex. mRNA ribosome complex. So in the last class I taught you the ribosome acts as a catalyst for peptide bond formation. So there is a particular site in the tRNA to recognize the ribosome. That I taught you in the last class. That is T loop I taught you. And here there are two subunits. Larger subunits of ribosome and smaller subunits of ribosome. So here the smaller subunits and the larger subunits. The smaller subunits consist of a binding site that can bind with the mRNA molecule. Suppose this is the mRNA molecule, or we can say the template strand, mRNA template strand. So this smaller subunit has a binding site for binding with the start codon of mRNA, which is the start codon AUG. So AUG is the start codon. Okay, that we taught you in the last class. So the larger subunits consist of 3 sites. So we can write over here, the larger subunits consist of 3 sites. They are E sites, P site and A site. Okay, so we can start with the A site. A site is the amino acid receiving site. So for the peptide bond formation, the amino acid is received by the A site of larger subunits. Okay. And there is a P site at the center, and this P site is the peptide or peptidase site or peptide bond formation site. Peptide bond formation site. That is the P site of larger subunits. And there is another site, that is E site, that is the exit site. So the tRNA, after binding the anticodon with the codon of mRNA, it gets released from the ribosomal complex through this E site that I will show you in the video. Okay, these are the three sites of larger subunits. And now we can see here the smaller subunits get bind with the start codon of mRNA. Then the tRNA, a charged tRNA with anticodon, which may be the anticodon, which is the anticodon it can bind with the UAC. So the tRNA, we can write the UAC tRNA. Okay. So the tRNA with UAC anticodon can base pair with the AUG or start codon of mRNA first. Okay. Then the ribosomal complex, the larger subunits get bind with the mRNA. Okay. So here the larger, after binding the tRNA with anticodon UAC on the AUG of mRNA, the larger subunits bind and form an mRNA complex. Okay. So this is the mRNA ribosomal complex. So first, the smaller subunits bind with the start codon of mRNA, or we can say the smaller subunits bind with the AUG codon of mRNA. Then the charged tRNA with anticodon UAC base pair with the start codon of mRNA. So there is a base pair. After that, the larger subunits get bind with the mRNA. Then there is a ribosome mRNA complex. Smaller subunits, larger subunits and mRNA. So there is a mRNA ribosomal complex will be formed. Is it clear? So this is the formation of ribosomal mRNA complex. Here this ribosome catalyzes the peptide bond formation. Actually, this tRNA with anticodon UAC consists of which amino acid? Methionine amino acid. So here at the amino acid receiving site of the tRNA, there is an amino acid. One amino acid is there. Okay. So the tRNA consists of an anticodon loop and an amino acid receiving loop at the top. So this tRNA consists of methionine amino acid. Okay, that's all about the initiation process. Next we can see the second step, elongation. So here now we can see the mRNA ribosomal complex AUG. Here the smaller subunits and larger subunits, and there is a tRNA with the anticodon UAC, and this is the larger subunits, this is the smaller subunits. So here this consists of methionine amino acid. Okay. So here elongation means the second amino acid or second charged tRNA bind with the next codon, suppose this is the next codon, U. So, the charged tRNA consists of A. Okay. So, here the next charged tRNA binds with the codon, next codon of mRNA. So, this is the mRNA site. So, next tRNA, charged tRNA with the anticodon, that is complementary to the codon of mRNA, bind with the mRNA codon. Okay. So, at that time, the first tRNA released from the ribosomal complex. When the second charged tRNA along with the amino acid molecule base pair with the mRNA codon, the first tRNA released from the ribosomal complex. So here, as I told you, there are three sites: exit sites, P site and A site. So the next charged tRNA enter into the A site of the ribosomal complex, and the first one released through the E site of the ribosomal complex. Okay. So, and next this ribosomes move from codon to codon along the mRNA. So, there is a movement of this ribosomal complex along the mRNA codons, and that results in the formation or adding of amino acid one by one. So here in the next tRNA another amino acid is there. So there is adding of another amino acid by the charged tRNA. So when it moves along the mRNA codon, there is amino acids are added one by one, and that results in the formation of peptide bond or polypeptide bond. Polypeptide bond. Is it clear? So actually the first tRNA, the carboxyl group of the first amino acid is connected to the amino group of the second amino acid. As I told you, the amino acids are added one by one and they are connected to each other. So, they are connected. This peptide bond is formed between the first amino acid and the second amino acid. And the carboxyl group of the first amino acid is connected to the amino group of the second amino acid. Thus, a polypeptide bond is formed by the elongation of this ribosome complex. One by one, amino acids are added one by the charged tRNA. When the third tRNA binds with the mRNA codon, the second one also released from the complex through the exit site, E site. Is it clear? Now the last point, that is terminations process. Termination. When this ribosome complex reaches the stop codon, which are the stop codon? UAA, UAG or UGA. So there must be a stop codon at the 3' end or 3' region. So if any of these three codons are there in the 3' area, the termination, or we can say the translation process, stops there. So the translation process or the synthesis of protein or the formation of polypeptide bonds stop at the termination site, whether it is having UAA or UAG or UGA codon in the mRNA. And there is a releasing factor. So stop codon. There is a releasing factor or release factor that helps to. So there is a formation of amino acid, that is the peptide bond formation by adding the charged tRNA along the mRNA, and finally when it reaches the stop codon, it gets dissociated from the mRNA by by this releasing factor. So we can say when it reaches the stop codon, the amino acid or the polypeptide bond get dissociated from the mRNA with the help of this releasing factor. And this is the translation process. I'll make you understand this process by showing a video. I'll show you the video. You just listen the video carefully.
It's found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl or P site and the aminoacyl or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
So this is actually translation process. One more point I want to tell you. See, as I told you, the mRNA acts as a template and the translation starts from 5' to 3' direction, and here the mRNA consists of AUG, a start codon, and a stop codon, UAG or UGA or UAA. A stop codon is there. So in between there is a peptide bond formation site, and there are some regions before the start codon, or we can say at the 5' region, or we can say before start codon. There are some region called UTR. They are called untranslated regions. Untranslated regions. That means translation do not occur in this area. There is another region that is after stop codon. That is the 3' region after stop codon. You can see these areas. So these areas there is no translation process, and after stop codon also there is no translation occur. So these areas are called untranslated region. The region on the mRNA strand before the start codon and after stop codon, and that region is called untranslated region because these regions do not take part in translation process, and that region or UTR is essential for the efficient translation process. That's all about translation process. Thank you.